Okay, so this video is going to be only over 4 slides, which I know is not very many, but it's because this experiment has popped up in sometimes with these qualifying exams, they'll specifically ask about this experiment in this section. So that's why I wanted to make a separate video. So the first part about DNA duplication, replication, and recombination is just how does it happen? You start with a double-stranded molecule. That double-stranded molecule has this double helix structure. You need to get a new molecule, and that new molecule needs to have both strands, and those strands also need to be in that double-stranded structure. And so how does that happen? Overall, it needs to be quick because, again, this is a very big, long strand of DNA, and so the entire thing needs to be replicated. It needs to be accurate because if you have mistakes, those are going to be mutations. And even if they happen only like once in 100 base pairs, for example, there's hundreds of thousands of base pairs. So you're talking about potentially thousands and thousands of mutations every single time that organism replicates its DNA. So it needs to be super accurate so that we don't make mistakes. And it needs to happen at the appropriate time in the cell cycle. Remember that this will occur. We will replicate the DNA during the S phase of the cell cycle prior to cell division. So the experiment that I'm referencing, the one that has happened in the past, is this group Meselson and Stahl. This was in the late 1950s, and they had an experiment that was allowed them to test 3 different hypotheses about how DNA replicates. And so these 3 hypotheses, you're going to start with your original double helix molecule, and the idea is that you take that original template molecule, and you're going to create an entirely new DNA molecule that is 100% new compared to the old one. And this is the conservative model, which is that your template strand is conserved so that the new copy that you make is 100% all brand new DNA. That's the conservative model. Semi-conservative model, well, actually, that's conservative. The dispersive model would be that you start with your template strand and then you have your new strand, but that both of these molecules that you create in the end are going to be a mixture of some pieces of old and some pieces of new. And that is dispersive, that like they're a mixture of both the new and the old. And then semi-conservative is in the middle. And this is the idea that, well, there's 2 strands in that original molecule, and the thing that you're going to produce has got 2 strands. So maybe one of the strands is old DNA and one of the strands is new DNA for both pieces. So with conservative, there's one that is entirely old and one that is entirely new. With semi-conservative, they both have half of the old and the new inside of them. They're both made of 1 strand of each. And then the dispersive says that both of the molecules are a mixture of all these little pieces that are just spread throughout. And in this figure, we're showing what that would look like through 2 rounds of replication, because what you're supposed to notice is that if it's conservative, the ratio of old DNA after the second round to new DNA would be like 1 to 3. There's 1 that is old and 3 that are new. If it was semi-conservative, the ratio of this mixture strand versus the new DNA strand would be like 1 to 1, because we got 2 and 2 of the 2 different combinations. And then if it's dispersive, it would be just like unpredictable. There'd be all these different pieces. And so every piece would be unique in terms of its ratio of old to new DNA. And so if we've got these 3 hypotheses and we got 3 different predictions about what you would see after 2 rounds of replication, that provides a model for an experiment. Because by the late 1950s, we had radio labeling that was available, where we can label a piece of DNA and it will be radioactive. And so they grew E. coli in a medium that had normal nitrogen, and then also some that had this heavy nitrogen, nitrogen 15. Remember that the atomic number of nitrogen is 7. And so you would expect that the nucleus would have 7 protons and 7 neutrons for an atomic mass of 14. So this one is like normal nitrogen, nitrogen 14. And then this nitrogen, nitrogen 15, that'd be like heavy nitrogen. It has an extra neutron. And so this is the radioactive one, and then this is like normal nitrogen. So by growing in these 2 different mediums, that means we're going to have E. coli that we have an ability to trace if they've got basically the purple DNA or the blue DNA. So they're going to allow the E. coli to divide and just live their life. They're going to replicate their DNA, but we're going to track how many generations there are. So after the first generation, that would basically be its first round of replication. And then when we get the second generation, because we're measuring how long they've been in this petri dish. We know how long it takes them to divide. So for the second round of replication, we would expect to see these sorts of ratios. And we're going to be able to see if it's purple or blue based on if they're labeled. And so in this experiment, this would be the outcome from the experiment. This is from the original paper. They've got light nitrogen, heavy nitrogen, and half heavy nitrogen. So the light nitrogen, this is the nitrogen that is like the nitrogen 14. It's normal nitrogen. The heavy nitrogen would be like the nitrogen 15. And then the half heavy, these are the pieces of DNA that have a mixture of both the old and the new inside of them. And so what they were able to see is that this is generations after N addition, right? So this is number of generations after they've added the normal nitrogen. And they can see that after one generation, because notice they did like increments here. So after one generation, DNA is a mix. It has got half heavy weight, like it's got one band and it's half heavy. Okay. So that means it would be like half purple, half blue. And so if you look across, you're like, okay, this one has two types of DNA. This one has one type of DNA. This one would have a whole bunch of different types of DNA. So the fact that we have only one band is in line with semi-conservative hypothesis. And then on top of that, this single band is the half heavy weight, meaning it's a mixture of nitrogen 14 and nitrogen 15. Well, with semi-conservative model of replication, that's what we would expect after the first round. Now we can go over to the second round of replication. So this is the first generation. Now we go to the second generation, which would be over here, 1.9. And by the time we get to about two generations, first of all, there's two bands, so two types of DNA. And then those two bands, some of them are light and some of them are half heavy. Remember that the light ones, those are basically the new DNA because they added nitrogen 14. So the light ones, those are the new DNA. So those in this picture, the nitrogen 14 would basically be like the blue one. Okay. And then the nitrogen 15 would be like the purple one. And so after two rounds, we would expect a 3-to-1 ratio. And when we go over here and we look at the bands, that's what we're seeing is we got a half heavy band and then we got a light band, which is again consistent with a semi-conservative model of replication. So the Meselson and Stahl experiment is able to demonstrate really cleanly that we have these three different hypotheses. We have predictions about what we would expect to see in a gel that has radio-labeled nitrogen. And then we're able to just look at the gel and say, okay, the gel is the same as which one of these predictions, and it was semi-conservative. So that's how we know that this is the way that DNA is replicated, is we have a template. That template is used to make the complement, the new DNA strand, and then each piece of DNA has equal parts of both. It has one template and one complement.